Alright, next, right, we'll be looking at how viruses cause diseases. Okay, so before I go into this part, right, just want you to refer to the learning outcomes at the front page of the notes, where you will find that this part, right, comes under a different topic, right? It's not particularly on viruses anymore. It's actually put under the topic of infectious diseases, extension topic A. All right. So for infectious diseases as a topic, right, we will look at it in more details next year, right? But I thought that because you are learning viruses, so it makes more sense to bring this particular learning outcome and put it under this set of notes, right? So it flows a bit better. You have learned about viruses, right? You have learned how it infects our cells, and now you learn about how such infections causes the cells to die and therefore diseases, right? So it flows a bit better. But still, right, next year when we learn about the infectious diseases, then I'll remind you of the link, right, and that this learning outcome, right, is going to be in this set of notes and not in the set of notes next year. Okay, I'll just make sure you remember this part. Yeah. Okay, so looking at how viral infections causes the host cells to die, right? And that will cause diseases because when the whole cells die, right, the cells are part of our tissue, right, and organs and so on, right? So when the whole cells die, then the function of the tissues and organs may be disrupted, and when the normal functions of the body is disrupted, and then that would be diseases lah, or that's how diseases arise. Okay, so the question is how does the viral infections results in the death of the whole cells, right? Okay, so one way is that viral infection, right, when the viruses infect the whole cells, right, the whole cells machinery is taken over, right, by the viruses to make viral proteins and viral genome. Right, so the whole cells would be unable to make its own proteins, right, which will be important for its function and also for its survival. Right, the raw materials like amino acids, right, energy in the form of ATP, right, all this would be depleted when the viruses just keep making copies of itself, right? So because of that, then the host is unable to make its own proteins, its own DNA or RNA, and therefore the whole cell is not able to function properly and also cannot survive. Okay, and when the viruses have assembled and they leave the whole cell, right, and that would usually or surely cause the cells to die. Okay, because if it is an envelope virus, remember that envelope viruses are released via budding, right? And budding involves the host cell surface membrane folding out and becoming the viral envelope, right? So when you have many viruses leaving via budding at the same time, what happens is the whole cell surface membrane will be depleted, right? Then the whole cell would burst, right? Loss of membrane and die lah. So for viruses that are not envelope viruses, they do not exit by budding. Usually they exit via the lysis of the whole cell, and therefore the whole cell die also. And when viral infections occur, right, obviously there would be the triggering of immune responses. And if you look at the reproductive cycle of the enveloped viruses, especially like influenza, HIV, right, one step is when the glycoproteins of the viruses are inserted into the whole cell surface membrane, right? So you can imagine our cells having viral glycoproteins on its surface, right? So when the immune cells come along, then the immune cells would recognize our own infected cells as a virus, right? Because they have viral glycoproteins, right? So the immune cells will then attack our whole cells, infected host cells, and the host cells will die lah. And as a defense mechanism, right, when our own cells are infected, our own cells would release something called cytokines to recruit the immune cells, right? So basically, the cytokines are like signaling molecules to the immune cells to tell the immune cells that, oh, I am infected. So then the immune cells would come and will kill the infected cells. Okay, but usually, right, what happens is there will be collateral damage, right? The immune cells would come and basically like kill the cells of one particular area. So even though maybe in this area only like 1 or 2 cells are infected, right, but more cells will be killed by the immune cells, right? So that causes quite a bit of damage to the tissues. Okay, so these are some general ways, right, how a viral infection may cause the death of infected cells and even neighboring cells as well. Okay. And for influenza, right, in particular, right, the influenza viruses infect the epithelial cells of the respiratory tract for a human, and such infection is specific, obviously, because of the presence of hemagglutinin, which recognizes specific receptors containing salicylic acid, right, found on these epithelial cells. Okay, so when the influenza viruses infect these epithelial cells of the respiratory tract, these cells die, as we have discussed in the previous slide, right? So when the epithelial cells die, right, then obviously they cannot function, right? The usual functions of such cells will be impaired. And so one of the function, right, is the clearance of mucus, right? Because these epithelial cells, right, have got a cilia on their surface, and so the movement of the cilia, right, actually sweep mucuses away from the respiratory tract. Okay, and mucuses function to trap things like dirt, dirty stuff, or other bacteria and viruses. So when such mucus is not removed, right, due to the death of the epithelial cells, right, then all these dirty stuff and bacteria and viruses get stuck in the mucus, which stays in the respiratory tract. Right, so the presence of such pathogens, right, would actually increase the immune response. And just now we looked at how sometimes immune response may cause collateral damage, right, and kill more cells, more of our own cells. Right, so when all these infectious agents and dirt are not cleared, right, the immune response would be actually quite severe and cause a lot of damage to our own tissues in the respiratory tract. Okay, another extension to this infection by influenza, right, is when certain bacteria are not cleared by the epithelial cells. They may cause secondary bacterial infections, which will result in pneumonia, right? Yeah, so that causes quite a bit of damage to the lung tissues. Right? There would be some release of a lot of fluid into the lungs, so the person basically drowned to death by its own body fluid, which is released into the lungs due to all this very severe damage to the lung tissues. That's pneumonia. That's the reason why, right? Sometimes when we are down with flu, right, and we see the doctor, and we should see the doctor, right? Sometimes the doctors would prescribe antibiotics, which, as you know, right, kills bacteria. Right, so even though it's a viral disease, right, the doctors sometimes prescribe drugs against bacteria. Why is that so? Right, so now that you have learned about how influenza infection may result in the lack of clearance of bacteria and also secondary bacterial infection, this should come as no surprise now. right? This is to prevent such secondary infection by bacteria, all right? And also, right, when any bacteria that are trapped, right, get killed because of the antibiotics, that also help to reduce the immune response and to reduce further damage to the cells and tissues of the respiratory tract that we have discussed just now. Okay? All right, now we look at HIV, right? So HIV infect the T helper cells, and the infection again would cause the T helper cells to die, and we have looked at the general mechanisms how the whole cells die, right? The first three here. Okay, but for HIV and T helper cells, right, there's like one more mechanism how infection can cause the cells to die, and that is through the formation of syncytium, which is when a few of the T helper cells fuse together. And the reason why is because when the infection occurs, right, one of the steps of the viral reproductive cycle is when the viral glycoproteins GP120 and GP41 are inserted into the cell surface membrane of an infected T helper cell, right? But T helper cells, unlike other cells like the epithelial cells, T helper cells are found in the blood. They can move, right? So a particular infected T helper cell with the glycoproteins from the viruses can move and come near another T helper cell. When that happens, then the GP120 can bind to the CD4 receptor and CCR5 co-receptor on another T helper cells, right? And remember what happens when GP120 binds? It causes fusion of membranes, right? But here, instead of fusion of viral envelope with the whole cell surface membrane, now it's the fusion between two cells, the fusion of the cell surface membrane between two cells, one infected cell and one neighboring cell that the infected cells has come near. Right, so what happens is when one cell is infected, such fusion with other cells may occur, and that actually kills more T helper cells, even though only one cell is infected. Right. So this is how syncytium formation, how the fusion between different cells may kill, well, more cells, more T helper cells. Okay. Right. So when the T helper cells die, right, and we have looked at how T helper cells are important for the immune system before, when we look at HIV, right, the immune system would be impaired, and therefore that give rise to AIDS, right, which is a lack of immune responses, right, to opportunistic infections, and we die by, well, some other infections. Okay, and HIV infection may also result in cancer as well. Reasons why is because when the viral DNA, right, is inserted into our human DNA, right, during the infection, it may cause cancer-causing mutations, right? So if it is inserted near a proto-oncogene, it may cause the proto-oncogene to be expressed more than it should be, like overexpression. Because when the viral DNA, which contains, of course, viral control elements like viral promoters, enhancers, and so on, and these promoters and enhancers may be more active than the human ones. So when the viral DNA is inserted near a proto-oncogene, that may cause the proto-oncogene to be overexpressed. Or if the viral DNA is inserted like in the middle of a tumor suppressor gene, then this tumor suppressor gene is disrupted, right? And that causes the proteins that is coded for by the tumor suppressor gene to become non-functional, right? So when you have overexpression of proto-oncogene or a non-functional tumor suppressor gene, right, then that contributes towards the development of cancer, as you have learned in cancer before. And when there is the HIV infection, which results in AIDS, right? The impairment of the immune system, right? One of the functions of immune system is not just to destroy pathogens, right? The immune system also recognizes and destroy cancer cells, or cells that are developing into cancer. Because cells that are developing into cancer, right, looks a bit different. They may have different proteins due to the mutations. So they are recognized as something that's foreign by the immune system. But when the immune system is impaired, then what is cancer cells or developing cancer cells are not going to be cleared by the immune system. So they stay around and develop further into full-blown cancer. Okay? And some viruses, like other viruses, may actually cause cancer as well, right? Like HPV, causing the human cervical cancer, right, for the females. So without the immune system, then such cancer-causing viruses may be able to more easily infect us and cause cancer. All right, so for HIV, right, because remember that they have this period in their reproductive cycle where the viral DNA is inserted into the human genome and it stays silently in the human genome. That's why we would observe a latent period in HIV infection. So it may be many years after the initial infection for the disease AIDS to arise, because the HIV viral DNA just stays inside the T helper cells. The T helper cells does not die, right? So there's no AIDS yet. But when the T helper cells are triggered, right, by certain other infections, then they would then transcribe, right, the viral DNA, and then the new viruses get made, and then the T helper cells die, and that's where we have AIDS. Right? So that may take actually like some years before that happen. Right? So this is the latent period in HIV infection. All right.